Okay, let's unpack this. For, well, millennia, humans have been tinkering with plants, right? From selecting the plumpest seeds in ancient fields
Mm-hmm.
to Gregor Mendel's, groundbreaking work showing how traits get passed down.
Mm-hmm.
We've always sought to improve the crops that sustain us.
Absolutely.
Imagine moving beyond just choosing the best natural variations, beyond what traditional breeding alone allows, to literally um, rewrite a plant's genetic code.
Indeed. And traditional breeding programs, while powerful, they can be incredibly slow. Plus, they're fundamentally limited by the existing genetic diversity within just one species.
Right.
The science we're diving into today, it offers a way to bypass those natural limits. It's really about harnessing the very building blocks of life, DNA, to introduce novel characteristics into plants, creating possibilities that were, well, once unimaginable.
Exactly. So today we're taking a deep dive into this fascinating world of recombinant DNA technology, plant biotechnology, and genomics.
That's the plan.
We're pulling our insights straight from a chapter in Raven Biology of Plants.
Yeah.
And our mission, it's to understand how these powerful tools are fundamentally reshaping the future of plants. From our big agricultural fields to the actual produce on our dinner tables and what it all means for you.
A really crucial area of modern biology.
Okay, so here's where it gets really interesting. Picture this.
Mm-hmm.
You can take a very specific gene from one organism and precisely insert it into another, maybe even a completely different species. That's the core, right, of recombinant DNA technology, a technique that popped up in 1973 and just completely changed the game.
You really did. What's truly fascinating here is that this technology lets geneticists engineer entirely new genetic combinations, new genotypes that simply wouldn't occur through natural breeding.
Yeah.
It's not just about the precision, though that's key. It's about enabling gene transfer between species that could never, ever hybridize naturally. It just unlocks this vast new world of genetic potential.
That sounds almost like molecular sorcery. How do scientists actually perform this cutting and pasting of DNA?
Yeah, sorcery is a good word for it sometimes. You're right to think of it that way. The process largely depends on these things called restriction enzymes.
Okay.
Think of them as molecular scissors, but like incredibly precise ones. They don't just cut anywhere. They recognize and snip double-stranded DNA only at very specific short sequences, almost like they're reading a secret code in the DNA. Often these sites are palindromic, reading the same forwards and backward on opposite strands.
And the sticky ends part, that's quite a vivid description. How do those work?
Right. Well, some restriction enzymes make clean, straight cuts across both DNA strands, but others, they make staggered cuts, a few nucleotides apart on each strand.
Ah, okay.
And this leaves a short, single-stranded overhang. These are our sticky ends.
Like little Velcro strips?
Exactly. Or imagine two puzzle pieces with complementary exposed edges. They can easily find and reattach to one another because their sequences match up. Crucially, any DNA cut by the same restriction enzyme will have compatible sticky ends. This is what allows DNA from completely different sources, say a bacterium and a plant, to be joined together seamlessly.
Wow.
You just need another enzyme, called DNA ligase, to sort of glue the backbone together. This property is really what makes almost unlimited genetic recombinations possible.
So, okay, once we have these sticky ends, how do we actually create a new modified genetic combination? Like put it all together?
Okay, so the basic procedure involves first isolating DNA from your donor organism, the one with the gene you want. Then you use a restriction enzyme to cut that DNA into fragments. Hopefully one fragment contains your gene of interest.
Got it.
These fragments are then mixed with small, circular, self-replicating DNA molecules. Usually these are bacterial plasmids.
Plasmids, right.
These plasmids act as tiny carriers, or vectors. Think of them as vehicles for ferrying the foreign DNA fragment into host cells, typically bacteria first.
Like a molecular delivery truck for genes.
Precisely. A tiny shuttle. So the plasmid, now carrying this inserted foreign DNA, is what we call recombinant DNA because it's a new combination.
Makes sense.
When bacterial cells then take up these recombinant plasmids, and not all of them do, those that succeed are considered transformed.
Okay.
As these transformed bacterial cells then multiply, they naturally replicate their own DNA, including the recombinant plasmid. So you get tons of identical copies of that foreign DNA fragment. This process, generating many identical DNA fragments, is known as DNA cloning, or sometimes gene cloning. Essentially, you've created a molecular factory just churning out millions of copies of a specific gene. The textbook's figure 10:3 shows this nicely. Plasmid cut open, foreign gene spliced in, bacteria take it up, bacteria multiply, copying the gene each time.
So you've got this molecular factory churning out millions of copies, but then the big challenge. How do you actually find the specific bacterial cells that successfully took up your target gene? Must be like finding a needle in a haystack, right?
It definitely can be, yeah. And that's where selectable marker genes and reporter genes come in. They're really clever tricks.
How do they work?
Well, for example, a really common approach is to engineer the plasmid. So it also carries a gene for resistance to an antibiotic, let's say ampicillin.
Yeah.
If we then grow all the bacteria transformed and untransformed on a growth medium containing ampicillin, well, only the cells that successfully took up the plasmid with this resistance gene will survive and grow.
So you just kill off all the ones that didn't get the plasmid.
Exactly. All the other cells simply die. It makes it pretty easy to select just the cells containing our recombinant DNA.
Clever.
Yeah.
And reporter genes are for, like, visual confirmation, right? You mentioned glowing plants.
Exactly. Reporter genes give us a clear visual cue. The lacZ gene is a classic example used in blue-white screening, which figure 10-4 illustrates.
Blue-white screening.
Yeah. If your foreign DNA successfully inserts into the lacZ gene on the plasmid, it disrupts that gene. Bacteria with an intact lacZ gene make an enzyme that turns a special sugar blue. But bacteria where the lacZ gene is disrupted by your inserted DNA, they can't make the enzyme, so they form white colonies.
So you just pick the white ones.
You pick the white ones because those are the ones that likely contain your recombinant plasmid with the inserted gene. Other reporters are cool too, like the green fluorescent protein, GFP, from jellyfish. You can fuse the GFP gene to your gene of interest. Then, when that protein is made in the plant cell, it glows green under UV light. Figure 10-5 shows this, tracking where an actin-binding protein is located because it's tagged with GFP.
Wow, you can literally see where it is.
Yeah. Or luciferase, the enzyme from fireflies that makes them glow. You can link its gene to a specific promoter, the on switch for another gene, so the plant will actually glow in the specific tissues or conditions where that promoter is active, like the tobacco plant shown glowing in figure 10-6. It's like turning on a tiny light to see exactly where and when a gene is working.
That's incredible. So if you want a whole collection of an organism's genes, not just one, you build a library.
That's right. A genomic library aims to contain cloned fragments representing most, if not all, of an organism's entire genome. Cut the whole genome up, clone all the pieces into vectors.
Okay.
Then there's something called a complementary DNA library, or cDNA library. This one's special. It's made from messenger RNA, mRNA, using an enzyme called reverse transcriptase.
From RNA back to DNA.
Exactly. And since it's based on the mRNA molecules actually being used by the cell at that moment, cDNA lacks introns, those non-coding sequences found in eukaryotic genes, and any other DNA that isn't transcribed. It mostly contains just the protein coding sequences.
Why is that useful?
Very useful if you want to express a eukaryotic gene in bacteria, because bacteria don't have the machinery to remove introns. cDNA gives you the clean coding sequence.
Makes sense. Now, for quickly making lots of copies of just one specific DNA segment, there's PCR, the polymerase chain reaction. I've heard this is incredibly powerful.
Oh, PCR is absolutely a game changer, a cornerstone of molecular biology labs everywhere. It's essentially like having a molecular photocopier specifically for DNA.
How does it work?
It works by cycles. First, you heat the DNA sample to a high temperature, like 95 degrees Celsius. This separates the two strands of the DNA double helix.
Melts it apart.
Right. Then you cool it down slightly, allowing short synthetic DNA sequences called primers to bind or anneal to the specific spots flanking the DNA segment you want to copy. These primers define the start and end points for copying.
Okay.
Then you raise the temperature a bit, typically to around 72 degrees Celsius. This is the optimal temperature for a special heat-resistant DNA polymerase enzyme, famously Taq polymerase.
Taq. I've heard of that. From hot springs.
Exactly. Discovered in a bacterium, Thermus aquaticus, living in Yellowstone National Park's hot springs. Because it can withstand the high temperatures needed to separate the DNA strands, it doesn't get destroyed during the heating cycles.
Ah, clever.
So Taq polymerase starts at the primers and synthesizes new DNA strands, complementary to the template strands. Now you have two copies of your target sequence where you started with one.
And you just repeat that.
You just repeat that cycle, denature, heat, anneal, cool, primers bind, extend, warm, Taq copies maybe 25 to 35 times. Each cycle doubles the amount of target DNA, so you get an exponential increase. Millions, even billions of copies from a tiny starting amount in just a few hours.
Incredible amplification.
Yeah.
So once you have all those copies, or maybe pieces from your library, you can figure out the exact sequence of nucleotides, right? The A's, T's, C's, and G's. That's DNA sequencing.
Precisely. That's the next step, figuring out the actual code. Initially, this was quite laborious. It often involved using different restriction enzymes to cut the DNA into various overlapping fragments.
Right.
Then you determine the sequence of each small fragment. A key technique here is gel electrophoresis, shown in figure 10-7, which separates DNA fragments based on their size. Smaller fragments move faster through the gel matrix when an electric current is applied.
Like a molecular sieve.
Exactly.
Yeah.
So you'd sequence all these overlapping fragments and then computationally piece them back together, like solving a complex jigsaw puzzle, to get the sequence of the original larger piece of DNA. Figure 10-8 kind of illustrates this overlapping fragment strategy.
Sounds painstaking.
It was. But now, thankfully, we have highly automated sequencing machines. They can sequence DNA much, much faster and on a much larger scale. This has made it feasible to sequence entire genomes, the complete genetic blueprint of organisms, and even compare genomes to find subtle variations within a species.
Okay, so we've talked about this incredible genetic toolkit, restriction enzymes, plasmids, cloning, PCR sequencing. What does all this mean for plants specifically? This is where plant biotechnology comes in, right?
Indeed. Plant biotechnology is essentially the application of all these genetic manipulation techniques, this whole toolkit, directly to plants. But its roots, interestingly, go back further than recombinant DNA.
Oh, yeah.
Yeah, you can trace its origins back to the mid-1850s, with early work on hydroponics figuring out exactly which mineral nutrients plants need to grow without soil.
Right, growing plants in nutrient solutions.
Exactly. That foundational research into plant nutrition eventually led to understanding plant hormones and their crucial role in growth and development. And that understanding paved the way for growing plant tissues and even whole plants in controlled lab environments.
Which brings us to that amazing idea of growing a whole plant from just a single cell. You mentioned totipotency earlier.
Yes, totipotency. It's a massive concept in plant biology. Plant tissue culture is the general technique of growing plant cells, tissues, or organs in a sterile, controlled environment, usually on a nutrient gel. A major application is micropropagation, also called clonal propagation. This is used to produce large numbers of genetically identical plants' clones very efficiently, often starting from just a few cells or a tiny piece of plant tissue.
Like making perfect copies.
Exactly. The underlying principle relies on totipotency. This is the remarkable, almost unique ability of many mature, differentiated plant cells to de-differentiate and then redevelop into an entire, fully functional plant given the right conditions and hormonal signals.
So even a leaf cell still has the instructions for making roots and flowers.
It does. This potential was hypothesized way back in 1902 by Gottlieb Haberland, but it was really famously demonstrated by F.C. Stewart and his colleagues in the 1950s. They took cells from a carrot root, cultured them, and managed to grow whole, fertile carrot plants from single cells.
Wow. From a root cell.
Yeah, and later others did it with tobacco pit cells. It proved that differentiated cells hadn't lost genetic information. They still held the complete blueprint. Figure 10-9 in the text gives a visual showing callus tissue, which is like an undifferentiated mass of cells derived from sugarcane. By tweaking the hormone levels in the growth medium, you can induce that callus to form roots and shoots, regenerating a whole plantlet.
And beyond just making lots of copies, you said it helps with plant diseases too.
Yes, very much so. Micropropagation is great for producing disease-free plants. Partly it's the sterile lab conditions, which obviously helps.
Sure.
But a key reason is that growers often culture the meristem or the very tip of the shoot. These are regions of rapidly dividing embryonic-like cells. And viruses, which can be a huge problem in vegetatively propagated crops, often can't invade these rapidly dividing tips very effectively. They move too slowly into these regions.
So the tips are often naturally virus-free.
Often, yes. So by culturing just these tips, you can generate whole plants that are free from viruses that might have infected the parent plant. This has been incredibly important for boosting yields and health in crops like potatoes, rhubarb, and many ornamentals that are typically cloned rather than grown from seed.
Okay, so tissue culture is powerful on its own. But then we get to actually introducing those foreign genes using the recombinant DNA tools. This is genetic engineering of plants, right?
Yeah.
And you mentioned it has big advantages over traditional breeding.
Absolutely. This is where we combine the tissue culture methods with the gene manipulation tools. The two main advantages of genetic engineering over traditional breeding are, one, the precision. You can often insert just a single, well-characterized gene,
Right. Much more targeted.
much more targeted, and two, the ability to transfer genes between species that simply could not interbreed naturally. You can take a gene from a bacterium, a fish, another plant species, and put it into your crop plant.
Opens up a whole new playbook.
Completely. It accesses a world of genetic potential that's totally unavailable to traditional breeders who rely on sexual compatibility. However, it's important to remember, after the initial gene transfer and regeneration of a transgenic plant, traditional plant breeding techniques are often still essential.
Well, why is that?
Well, you need to make sure the inserted gene, the transgene, is stably inherited by the offspring. And you often need to cross the transgenic plant with elite crop varieties to combine the new trait with all the other desirable agricultural characteristics like high yield, good taste, regional adaptation. So it's often a combination of the new tech and traditional methods.
That makes sense. So how do scientists actually get those new genes into the plant cells? You mentioned vectors earlier for bacteria, but how do you deliver DNA to a plant cell?
Great question. One of the most widely used and frankly elegant methods uses a natural genetic engineer. The soil bacterium, Agrobacterium tumefaciens.
Agrobacterium. I think I've heard of that. Doesn't it cause tumors in plants?
It does. It causes a disease called crown gall, where you see these tumor-like growths often near the soil line on the stem. Figure 1010 shows an example on a tomato stem.
So how is a disease-causing bacterium useful?
Well, the way it causes the tumor is fascinating. The bacterium contains a special plasmid called the tumor-inducing, or T plasmid. Figure 1011 diagrams this plasmid. Agrobacterium has the natural ability to cut out a specific segment of this T plasmid called the tDNA, for transfer DNA, and transfer it directly into the plant cell's nucleus, where it gets integrated into the plant's own chromosomes.
It injects its DNA into the plant's DNA.
Essentially, yes. The tDNA region carries genes that, once inside the plant cell, basically hijack the plant's machinery. They force the plant cell to produce unusual amino acids called opines, which the bacteria use as food, and they also make the plant cells divide uncontrollably, forming the gall or tumor.
Wow. Nature's own genetic engineer.
Exactly. So scientists realized they could harness this natural delivery system. They learned how to disarm the T plasmid.
Disarm it.
Meaning, they remove the genes within the tDNA region that cause tumor formation and opine production, but they leave the border sequences that define the tDNA and the bacterial genes, Vir genes, also on the T plasmid, needed for the transfer process itself.
Ah, so they take out the bad parts.
Right. And then, into that engineered tDNA region, they splice the gene or genes they want to introduce into the plant, say a gene for insect resistance or herbicide tolerance.
Over.
Then they put this modified T plasmid back into Agrobacterium and co-culture the bacteria with plant cells or small pieces of plant tissue, often using tissue culture methods. The Agrobacterium does its natural thing. It transfers the engineered tDNA, carrying the desired foreign gene, into the plant cell's genomes.
And then you use tissue culture to grow those modified cells into a whole plant?
Exactly. You select for the transformed cells, often using marker genes, just like in bacteria, and use plant hormones to regenerate them into complete transgenic plants. These plants contain the foreign gene integrated into their own DNA. And importantly, as shown in figure 1012, they usually pass this transgene onto their offspring through seeds, following standard Mendelian inheritance patterns.
It's incredibly elegant, hijacking nature's system. Are there other ways to get genes into plants, maybe if Agrobacterium doesn't work well with a certain species?
Yes, definitely. Agrobacterium works great for many broad-leafed plants, dicots, but it was initially trickier for grasses like corn, rice, and wheat, monocots, although methods have improved. So other techniques were developed. One is electrophoresis.
Electrophoresis. Sounds electric.
It is. You first need to remove the plant cell's rigid cell wall, creating what's called a protoplast, basically a naked plant cell. Then you suspend these protoplasts in a solution containing the DNA you want to introduce, and you apply brief, high-voltage electrical pulses. Pretty much. The electrical pulse creates temporary, tiny pores in the cell membrane, allowing the DNA from the solution to enter the cell before the membrane reseals. Then you have to culture these protoplasts carefully to regenerate the cell wall and eventually a whole plant.
Okay. And the other one? You mentioned a gene gun.
Right. Particle bombardment, also known as biolistics, or more colloquially, the gene gun. This is a physical method. You coat microscopic beads, usually made of gold or tungsten, with the DNA or sometimes RNA that you want to deliver.
Tiny bullets coated with genes.
Essentially, yes. Then you use a device that uses high pressure, like helium gas, to literally shoot these DNA-coated microprojectiles at high velocity into plant cells or tissues.
Shoots them right through the cell wall.
Yep, penetrates the cell wall and membrane. Some of the DNA gets dislodged from the beads inside the cell and can get integrated into the plant's genome. It sounds crude, but it can be effective, especially for plants resistant to agrobacterium transformation.
Any famous examples of the gene gun working?
Absolutely. A classic success story is how it was used to rescue the Hawaiian papaya industry back in the 1990s.
Oh, yeah. What happened?
The industry was being decimated by the papaya ring spot virus, PRSV. It was devastating. Scientists used the gene gun to shoot genes encoding the virus's own coat protein into papaya tissue explants.
The virus's own gene.
Yes. The idea, borrowed from earlier work, was that expressing the coat protein gene in the plant could somehow interfere with the virus infection cycle, conferring resistance, a kind of genetic immunization.
Did it work?
It worked beautifully. They regenerated transgenic papaya plants expressing the coat protein gene, and these plants showed remarkable resistance to PRSV infection, as you can see in Figure 10.13, comparing a resistant transgenic plant to a susceptible non-transgenic one. This technology essentially saved the commercial papaya industry in Hawaii.
Wow, that's a direct impact. So these two methods, agrobacterium and the gene gun, are the main ways.
Yes, agrobacterium-mediated transformation and particle bombardment are currently the most widely used and successful methods for creating genetically engineered plants.
Okay, so the tools are powerful, the delivery methods are clever. These applications sound incredibly diverse, really tackling some massive challenges. What are some of the biggest impacts we've actually seen out in the fields, on the ground?
The impact on agriculture has been truly significant, yeah? Let's take insect resistance, for example. This is a big one. Scientists took genes from a common soil bacterium, Bacillus thuringiensis, or beet. This bacterium naturally produces proteins that are toxic to certain insect larvae, like caterpillars, but are harmless to mammals and most other organisms.
Oh, beet. I've heard of that in organic farming, too.
Exactly. Organic farmers sometimes spray beet spores as a biological insecticide. Genetic engineering takes it a step further. The gene encoding the beet toxin protein is inserted directly into the crop plant's genome.
So the plant makes its own insecticide.
Precisely. Crops like maize, corn, and cotton have been engineered to produce beety proteins. So when a susceptible insect larva, like the European corn borer munching on corn stalks, like in Figure 10.14a, or the cotton bollworm attacking cotton bolls, eats the plant tissue, it ingests the beety protein, which disrupts its digestive system, and the insect dies.
That must dramatically reduce the need for spraying chemical insecticides.
Hugely. It's been a major benefit for farmers, reducing costs and labor, and also for the environment by lowering the amount of broad-spectrum chemical pesticides used. There are also benefits for non-target beneficial insects.
But is there a catch? You mentioned resistance earlier.
There is a potential downside, yes. Just like with chemical insecticides, if you rely too heavily on a single mode of action, insects can evolve resistance over time. And unfortunately, resistance to biotoxins has been reported in some populations of insects, like pink bollworm in India.
So it's not a permanent silver bullet.
Not necessarily. It requires careful management strategies like planting refuges of non-brute crops nearby to slow down the evolution of resistance. It's an ongoing challenge.
Okay, that's insect resistance. What about dealing with weeds? That's another huge issue for farmers.
Absolutely. Another major application is engineering crops for herbicide tolerance. Many widely grown crops like soybeans, corn, cotton, and canola have been genetically modified to tolerate specific broad-spectrum herbicides, most famously glyphosate, the active ingredient in Roundup.
Roundup-ready crops.
Exactly. The engineered crops have a modified version of a key plant enzyme, EPSP synthase, that isn't affected by glyphosate, or they have an extra gene that allows them to break down the herbicide.
And so the farmer can spray glyphosate over the entire field.
And it kills the weeds, but the crop plants survive unharmed. This simplifies weed management significantly. It has also facilitated the adoption of no-till or conservation tillage farming practices.
How does it help with no-till?
Because farmers can control weeds chemically without needing to plow or till the soil, they can leave crop residues on the surface. This is fantastic for reducing soil erosion by wind and water, conserving soil moisture, and keeping carbon stored in the soil rather than releasing it into the atmosphere. So big environmental benefits there.
But again, The resistance issue.
Yes, similar challenge. The widespread and repeated use of glyphosate, partly driven by the prevalence of glyphosate-tolerant crops, has unfortunately led to the evolution of glyphosate-resistant weeds in many parts of the world.
Superweeds.
Well, super might be a strong word, but certainly weeds that are much harder to control with that specific herbicide, requiring farmers to use other herbicides or integrate different weed management tactics. It highlights the need for diverse strategies.
Okay, insect resistance, herbicide tolerance. What about reducing waste, especially after the crops are harvested? Food spoilage is a huge problem.
It is, and genetic engineering can also be used to manipulate plant growth and development processes, including those related to spoilage. A classic example involves fruit ripening.
Tomatoes, right?
Tomatoes were an early target, yeah. Ripening is largely controlled by the plant hormone ethylene. Scientists identified genes involved in ethylene synthesis and were able to engineer tomatoes where ethylene production was significantly reduced.
So they ripen much more slowly.
Exactly. Figure 1014b shows a comparison. This is potentially very useful for fruits that need to be shipped long distances. They can be harvested when mature, but still firm, shipped, and then perhaps treated with ethylene gas at the destination to trigger final ripening just before they go on sale.
Less spoilage in transit.
That's the idea. Similar approaches have been used to delay flower wilting, for instance, in petunias, shown in figure 1014c and d. By blocking ethylene action, you can extend the vase life of cut flowers significantly, which is obviously huge for the floriculture industry.
Makes sense.
And it's not just ripening or wilting. You can target senescence or aging in general. For example, by manipulating genes involved in the production of another plant hormone, cytokinin, which tends to delay aging, researchers have created tobacco plants where the leaves stay green and functional much longer, as shown in figure 1014e. This could have implications for forage crops or leafy vegetables.
Fascinating. So, controlling pests, weeds, spoilage. But what about improving the actual nutritional quality of the food we eat? Can biotechnology help there?
Yes, and this is an area with enormous potential impact on human health. There have been successes in altering the fatty acid profiles of oils produced by crops like soybeans and canola, aiming to reduce unhealthy saturated fats and increase healthier unsaturated fats.
Healthier cooking oils.
Potentially, yes. There are also efforts to modify starch content or composition in crops like potatoes, maybe leading to French fries that absorb less oil when cooked, for instance.
Healthier fries. Signing up.
Huh. We'll see. But perhaps the most famous example of nutritional enhancement is golden rice. You see it in figure 10:1, the very first figure in the chapter.
Golden rice, right? Vitamin A.
Exactly. Regular rice grains contain almost no beta-carotene, which our bodies convert into vitamin A. But vitamin A deficiency is a massive public health problem in many parts of the world, especially southern Asia, causing hundreds of thousands of cases of childhood blindness and contributing to many deaths each year.
It's a huge issue.
It is. So scientists engineered rice to produce beta-carotene in the endosperm, the part of the grain that people eat. They did this by introducing a couple of genes, one from daffodil and one from a bacterium, that together complete the biochemical pathway for beta-carotene synthesis.
And the rice turns yellow, or golden.
Right, hence the name golden rice. The potential health benefits are immense, providing a vital nutrient directly through a staple food crop for populations who need it most.
Has it been widely adopted? I feel like I've heard about controversies.
There have definitely been challenges and controversies. Getting regulatory approval in different countries took a long time. There were also initial challenges in breeding the golden rice trait into locally adapted red varieties that farmers actually want to grow and consumers want to eat. But progress continues to be made, and the potential for saving sight and lives remains enormous. It really highlights both the promise and the complexities of this technology.
Beyond food, are there other products being made in engineered plants?
Yes, definitely. Plants are being explored as bioreactors or biofactories to produce valuable substances that might otherwise be difficult or expensive to make.
Like pharmaceuticals.
Exactly. Researchers have engineered plants, like tobacco, to produce pharmaceutical proteins, such as human growth hormone or antibodies. The idea is you could potentially grow large amounts of these proteins relatively cheaply in plants and then extract them.
Plant-made medicine.
That's the vision. There's also research into producing industrial materials like biodegradable plastics, polyhydroxyl alkanoates or PHAs, in plants such as poplars. So the applications really extend beyond just food and feed.
Okay, so these are incredible advancements, no doubt, offering potential solutions to really massive global challenges. Pests, weeds, spoilage, malnutrition, even producing medicines. But with such powerful tools, What about the potential downsides or risks? You touched on resistance, but what else?
That's a really important and often debated question. First, regarding food safety. Based on decades of research and consumption, there's currently no credible scientific evidence that the process of gene transfer itself, or the genetically modified foods currently approved and on the market, pose any direct risk to human health compared to their conventionally bred counterparts.
So the food itself is considered safe by scientific consensus.
Broadly, yes. The focus should be on the specific new trait introduced, not the method used to introduce it. Every new crop variety, whether from conventional breeding or genetic engineering, needs evaluation. But the process of genetic engineering hasn't been shown to introduce unique health risks.
Okay. What about environmental risks then?
That's where the concerns are perhaps more prominent. A primary worry is the potential for gene flow, the modified genes escaping from the cultivated crop into wild or weedy relatives through natural cross-pollination through hybridization. If, for example, a gene for herbicide tolerance were to transfer into a closely related weed species, you could potentially create a superweed that's resistant to that herbicide, making it much harder to control.
Has that happened?
The potential exists, particularly in regions where crops have compatible wild relatives growing nearby. The likelihood and impact depend heavily on the specific gene, the crop, the environment, and the presence of those relatives. It requires careful risk assessment and management on a case-by-case basis. For many major crops in places like North America, compatible wild relatives are rare, making the risk lower. But it's not zero everywhere.
So it's a possibility that needs careful watching.
Absolutely. It needs to be considered in the environmental context. It's also crucial to remember, as we discussed, the potential for developing resistant insects or weeds due to selection pressure from the engineered traits themselves. These are ecological considerations.
Right.
And overall, it's important to view genetically modified crops not as a magic bullet, but as one tool among many in the pursuit of sustainable agriculture. They need to be integrated thoughtfully with other approaches like traditional breeding, developing new crop varieties, using biological controls for pests, and improving cultivation practices like crop rotation and integrated pest management.
A multifaceted approach, not just relying on one technology.
Okay, exactly.
Okay, let's shift gears slightly. Moving from tinkering with individual genes to looking at the entire instruction manual. Let's talk about genomics. What's the broad objective of this field? Sounds huge.
It is huge. Genomics is the comprehensive study of entire genomes. It looks at their complete genetic content, how all that information is organized on the chromosomes, how the genes function collectively, and how genomes evolve over time. It's really the ultimate extension of recombinant DNA technology, moving from single genes to a global analysis of all genetic information in an organism.
Okay, so within genomics, there's structural genomics.
Yeah.
Is that about mapping out where all the genes physically are, like drawing a detailed blueprint of the chromosomes?
Precisely. Structural genomics focuses on deciphering the organization and the exact sequence of the genetic information within a genome. This involves creating different kinds of maps. Yeah. There are genetic maps, which are based on recombination frequencies, how often genes located on the same chromosome get separated during meiosis, the process of making egg and sperm cells. This gives you a relative order and rough distance between genes, measured in map units or centimorgans.
Okay, a rough guide.
A rough guide, yes. Then there are physical maps, which are much more precise. These are based on the direct analysis of the DNA molecule itself, usually through sequencing. They show the actual physical locations of genes and other DNA sequences on the chromosomes, with distances measured in the number of base pairs, BP, or kilobases, KB.
So much higher resolution.
Much higher resolution. Figure 1015 in the textbook gives a nice visual comparison showing how a genetic map and a physical map of the same chromosome region might look. They provide complementary information about the genome's structure. The ultimate physical map, of course, is the complete DNA sequence of the entire genome.
Once we have that map, that sequence,
Yeah.
then we need to figure out what everything actually does, right?
Yeah.
Is that functional genomics?
That's exactly the aim of functional genomics. You've got the blueprint from structural genomics. Now functional genomics tries to understand what all those parts do. Its goals are to identify all the genes within the sequence, determine when and where they are expressed or turned on during organism's life cycle or under different conditions, and ultimately uncover the function of the proteins or RNA molecules those genes produce.
Because just having the sequence isn't enough.
Not at all. Knowing the sequence is just the first step. We need to understand its biological meaning and purpose. How does functional genomics tackle this?
Yeah, how do you figure out function?
Well, a common first approach, once you've identified a potential gene sequence, is a homology search. You use computer algorithms to compare your newly discovered gene sequence against massive public databases that contain sequences of genes whose functions are already known from many different organisms.
Looking for similarities.
Exactly. If your unknown gene sequence shows significant similarity homology to a gene with a known function, say an enzyme involved in photosynthesis,
Yeah.
it provides a strong clue, a tentative hypothesis about your gene's function.
Okay, that makes sense. What else?
Another powerful method involves creating and studying knockout mutants. Here, scientists specifically inactivate or knock out a single target gene in an organism.
Turn it off.
Right. Then they carefully observe the resulting changes in the organism's appearance, its phenotype, its physiology, or its biochemistry. By seeing what goes wrong or changes when the gene is missing, you can infer the gene's normal role or function.
Learn by breaking it.
In a controlled way, yes, it's
Yeah.
a very common strategy.
And what about knowing when and where genes are active? Not just what they do, but their activity patterns.
Great question. That's crucial for understanding development and responses to the environment. For that, we use techniques that measure gene expression on a large scale. One key technology here is DNA microarray analysis, sometimes called gene chips.
Gene chips. I've heard that term. How they work?
Imagine a small glass slide, maybe the size of a microscope slide. This chip is covered with thousands, even tens of thousands of microscopic spots. Each spot contains many copies of a known short DNA sequence representing a specific gene from the organism you're studying. So you might have spots for nearly every gene in the genome on one tiny chip.
Wow, the whole genome on a chip.
Many of its genes, yeah. Then you take the RNA from the cells or tissue you're interested in, say, leaf cells under drought stress versus normal conditions. You convert this RNA into labeled cDNA, often fluorescently labeled.
Okay.
Then you apply this labeled cDNA mixture to the microarray chip. The cDNA molecules will hybridize or bind only to the spots on the chip containing their complementary DNA sequence.
So stick to their matching gene spot.
Exactly. You wash away any unbound cDNA, and then you use a scanner to detect the fluorescence signal from each spot. The intensity of the signal on a particular spot tells you how much mRNA corresponding to that gene was present in your original sample.
So the bright spots are the highly active genes.
Correct. By comparing the patterns of bright and dim spots between different conditions, like drought versus normal, you can see which genes were turned up, turned down, or stayed the same in response to that condition. It gives you a snapshot of the activity of thousands of genes, simultaneously a global view of gene expression.
That's incredibly powerful for understanding complex responses.
Absolutely. It lets us study gene expression changes during development in different tissues or in response to all sorts of environmental stresses like drought, heat, pathogens, nutrient levels, and so on.
Okay, so we have structural and functional genomics. What about comparative genomics? That sounds like comparing blueprints between different species.
That's precisely what it is. Comparative genomics compares the gene content, the gene functions, and the overall organization and structure of genomes across different species, sometimes closely related, sometimes very distantly related.
What do we learn from that?
We learn a tremendous amount about evolution. By comparing genomes, we can identify genes that are conserved across vast evolutionary distances, suggesting they perform fundamental life functions. We can see which genes are unique to certain groups, contributing to their specific characteristics. We can track how genomes have changed over time through processes like gene duplication, gene loss, rearrangements, and even the acquisition of new genes.
So it helps build the tree of life.
Absolutely. Genome comparisons have provided incredibly strong support for and refined our understanding of evolutionary relationships. For instance, comparing whole genomes powerfully confirmed the existence of the three fundamental domains of life: bacteria, archaea, and eukarya, showing that each domain has many unique genes, while also sharing a core set of essential genes, especially those involved in basic cellular processes like DNA replication and protein synthesis.
Fascinating. So looking at genomes also tells us about the diversity of life at that fundamental level.
Immensely so. Prokaryotic genomes, for example, bacteria and archaea, show incredible diversity. Their genome sizes can vary hugely. There's also significant genetic diversity within what we call a single species, and they frequently engage in horizontal gene transfer.
Horizontal transfer, not parent to offspring.
Right. Bacteria can exchange genetic material directly between individuals, even if they are distantly related species. This allows for rapid adaptation and the spread of traits like antibiotic resistance.
Wow. What about eukaryotes, like plants and animals?
Eukaryotic genomes are generally larger and more complex than prokaryotic ones, often containing large amounts of non-coding DNA, including introns within genes. But interestingly, the number of protein-coding genes doesn't always directly correlate with what we perceive as an organism's biological complexity.
Really. More complex doesn't always mean more genes.
Not necessarily. Figure 1016 provides some examples. The simple mustard weed, Arabidopsis thaliana, which is a major model organism for plant research, has about 27,000 genes. Humans have roughly 20,000, 25,000. Rice has even more genes than Arabidopsis, maybe around 30,000, 40,000, depending on how you count.
Why would plants have so many genes?
Often it's due to past events of gene duplication or even whole genome duplication, polyploidy, which is quite common in plant evolution. These duplications provide raw material for evolution. One copy can retain the original function while the other copy is free to mutate and potentially acquire a new function over time.
Extra copies for innovation.
Kinda, yeah.
Wow.
And the knowledge gained from sequencing these plant genomes, especially key model organisms and major crops, is incredibly valuable. Arabidopsis was chosen as a model because it has a relatively small genome for a plant. It grows very quickly, produces lots of seeds, and is easy to manipulate genetically.
A lab workhorse.
Exactly. Rice, or Rhiza sativa, was another early target because it's arguably the world's single most important food crop, feeding billions. Plus, being a grass, understanding its genome provides insights that can often be applied to other vital cereal crops like wheat, maize, corn, barley, and sorghum because they share a common evolutionary history and many related genes.
So understanding rice helps understand corn and wheat, too.
To a large extent, yes. By comparing genomes and using functional genomics tools, researchers can identify genes responsible for important agricultural traits, things like yield potential, grain quality, tolerance to drought or salt stress, resistance to diseases and pests. And once identified, these genes become targets for improvement, either through advanced breeding techniques or potentially through further genetic engineering.
Bringing it all back to improving our crops.
Ultimately, yes, that's a major driving force behind much of this research.
So to wrap this up, what does this all mean for you, the listener? We've taken quite a journey from, ancient farmers saving the best seeds all the way to the incredible precision of modern molecular biology.
It's a huge leap.
Recombinant DNA technology gives us these amazing tools like molecular scissors and delivery trucks for genes. Plant biotechnology shows us the practical applications, creating plants resistant to pests, tolerant to herbicides, with delayed ripening or enhanced nutrition. And genomics helps us understand the entire blueprint of life, the whole instruction manual, accelerating our ability to pinpoint the genes responsible for almost any biological phenomenon.
It really provides an immensely powerful set of resources, not just for crop improvement, but also for fundamental biological discovery, understanding how life works at the molecular level, and unraveling complex evolutionary relationships. It truly highlights how deeply interconnected the molecular world is with the macroscopic world we see, our food supply, our health, and the environment.
Absolutely. From, those glowing tobacco plants we talked about, to the virus-resistant papayas that saved an industry, and the incredible promise, despite the challenges, of golden rice. This deep dive really shows us a world where the very nature of plants is being understood and, in some cases, reshaped at a fundamental level. It's honestly a testament to human ingenuity, and it offers a compelling, if complex, glimpse into the future of food, medicine, and maybe even the health of our planet.
And thinking forward, it does raise a really important question for all of us. As our ability to understand and modify life at this fundamental level continues to grow so rapidly, how will we as a society choose to apply these incredibly powerful insights? How can we use them wisely and ethically to help solve pressing global challenges like food security and climate change and ensure a sustainable future for everyone?
Indeed. That is definitely a thought to chew on. Thank you so much for joining us on this incredible deep dive into the world of plant genetic engineering, biotechnology, and genomics. We really hope you've gained a new appreciation for the amazing science that's literally growing our future.
Thanks for tuning in.